Assalamu alaikum. Lysosomes and proteasomes. The lysosome is an enveloped organelle in the cell. It is basically a vesicle that contains hydrolytic enzymes inside, and its main function is to break down substances. It contains a lot of degradative enzymes, all present inside this lysosome. And since it contains all these degradative enzymes, its membrane keeps them all inside so that they do not cause any damage to the cell by mistake, because if they are released, the cell will be destroyed. And this is called autolysis, meaning that the cell destroys itself. Lysosomal enzymes are synthesized in the endoplasmic reticulum, but they only work in an acidic medium with low pH. And this is what makes the lysosome special, since it provides an acidic environment for these enzymes to work. The lysosomes dispose wastes, and they work directly with the endosomes that come from the outside of the cell and mix with the lysosomes to break down their components. Lysosomes also break down foreign bodies in the phagosome, such as bacteria and dead cells. Since lysosomes have many hydrolyzing enzymes, this makes them of special shape in histology, as they appear under the microscope as densely packed particles. And it must be mentioned that the number of lysosomes in the cell varies according to the function of the cell. As you can guess, they are present in large numbers in white blood cells that fight foreign bodies and phagocyte dead cells. But in general, lysosomes are present in all human cells. That was some information about the lysosomes, and we will learn more in biochemistry, metabolism, and pathology. Now let's move to the proteasome. It is a complex of proteins present in the cytoplasm. Its function is to degrade damaged and misfolded proteins in the cell, and it is not enveloped with a membrane. Therefore, it may or may not be considered as an organelle of the cell. Proteasome breaks down proteins in the following cases. If a particular protein is present in abundance and its number is sufficient, it breaks down any new proteins that is synthesized of this type to prevent the accumulation of proteins. If the protein is damaged, mutated, misfolded, or not modified properly. Proteasomes break down the proteins into amino acids, which are then reused to make other proteins. It is also important to know that the proteasomes use ATP as an energy source to accomplish their process. For proteasomes to recognize a protein that needs to be degraded, certain enzymes tag this protein with a special small protein called Ubiquitin, which indicates that this protein attached to it has been targeted and the proteasome can recognize it. Too much information for two tiny organelles that are only good at destroying. Now let's do a quick review to the main information. The lysosome is a membrane-enveloped organelle that contains hydrolytic enzymes inside. Its function is to degrade substances, from disposing of waste to destroying foreign particles. Lysosomes receive their enzymes from the endoplasmic reticulum and provide them with an acidic environment with a pH of 5 suitable for the degrading enzymes to work. The number of lysosomes varies according to the function of the cell, and they are abundantly present in some white blood cells. As for the proteasome, it is a protein complex not enveloped with any membrane that breaks down proteins tagged with ubiquitin into amino acids that can be reused to make other proteins. This is to either get rid of the accumulated proteins or from the denatured ones. This concludes our discussion about the subject. If you want to test your knowledge by solving the questions, continue the video. Lysosome provides an acidic environment for its enzymes to function, meaning that the pH is less than 7, which is about 5. The proteasomes break down unneeded proteins, regardless of their size. We said that the proteasome breaks down proteins into amino acids to be reused in the synthesis of other proteins, but it does not break down into atoms.